now. So this is material from Chapter 2. It should have sounded familiar for those of you who already looked over Chapter 2, because it's talking about basic chemistry like atoms, molecules, different types of bonds, covalent, ionic, non-covalent, those types of bonds, and some biochemistry as far as introducing or reviewing the organic macromolecules and the interactions, so the assembly and disassembly of these macromolecules from their subunits. Okay, so the smallest unit of matter, raise your hand, please. Giuliano? Atoms, right? So this is the basic unit, not of life, right? Because atoms make up both inanimate and animate objects. But it's the smallest unit that gives the molecules those chemical properties, right? So it's the smallest unit that defines the chemical properties of that element. Okay, so molecules are combined atoms, right? So one or more elements make up molecules. And they can be bound together in 3 basic ways, which include, Giuliano? Okay, so what do we call these? Covalent, yes. Covalent and ionic. And when they're macromolecules, there are weaker interactions that hold them together. Aileen, can you help us with some other? Non-covalent bonds. Hydrogen bonds, van der Waals, yep. Electrostatic interactions, so charged interactions, exactly. Okay, and we already went over the 4 macromolecules last time, so look back in your notes. What are those 4 categories? Quickly. Yep. Carbohydrates, lipids. Jesse? Proteins, yep. Nucleic acids. Thanks, Khaled. Okay, so the atoms have the basic structure of a nucleus. Do not confuse this nucleus with the membrane-bound organelle found in cells. This is what? Bigger or smaller? Much, much, much smaller, right? Subatomic. So there's a tiny concentration in the middle of each atom called the nucleus that contains what part of the atom? Or what parts of the atom? Yep, Aileen? Protons and neutrons. And what's kind of spinning around that in a cloud of? Yep, electrons, right. So just to look at a couple of atoms, who can take a guess at what these atoms are? This one is simple, right? Hydrogen, right? What's this one? Carbon. How did you know that? Right, 6 electrons, 6, what are the positively charged ones? Protons, and the ones that don't have a positive or negative? Neutrons, right? So the atomic number of each atom is written in the periodic table of elements at the top. What does that represent? Just protons, yeah. What about the atomic weight, which is at the bottom here? Protons plus neutrons, right? So that cloud of electrons, it's really, you can think of it like a cloud. It's not really anything you can grab onto or has any weight. If you walk through a mist or fly through a cloud in an airplane, you know, that doesn't really give any significant weight to the atom. It's just the stuff in the nucleus that gives that weight. And isotopes will have a different number of what part? Neutrons. Yeah. will determine the isotopes. So one of the ways that we can tell the age of something is by measuring carbon-14, which decays very slowly over time. Okay, so the electrons are what can tell us a lot about the reactivity of atoms and how they will form bonds. So in this chart book, you can see all the red dots represent an electron shell that is not complete. So if the electron shell is not complete, is the atom stable or unstable? Unstable, unhappy, and it wants to either share electrons, so is ionic or covalent bonding sharing electrons? Covalent bonding is sharing electrons. And ionic bonding is when you actually have the transfer. So one atom is giving up their electron, the other one is gaining it in order to achieve that happy balance of a full electron shell. Okay? Okay, so ionic bonding, such as sodium with chloride, will lead to ions. Okay, so the sodium is donating this one extra electron it has in its third shell. Then it's happy because it has this full second shell. And the chloride ion, or the chloride atom, which is just one electron short of having a full third shell, will gain that electron and become a chloride ion. So you should be able to draw an atom in this manner where you have the electron shells showing. But there also in your book, it includes other ways of representing these molecules. So covalent bonding, the sharing of the electrons, is that going to be a stronger bond or a weaker bond than ionic bonds? Alina? Stronger. So they're actually, the interactions between the atoms will reach a balanced point where you can actually measure the distances between these points through things like X-ray crystallography, to the point where they're stable and strong. The three basic elements that make up most of organic molecules, most of the molecules you find in living cells, are nitrogen oxide. What is missing here for the other major? Hydrogen, phosphorus, sulfur, right? So did anyone learn the chunks? Okay, to remember the 6 basic elements that are involved in macromolecules or living molecules associated with living cells. Water, I've shown here, or your book shows here, in the ball and stick model, with red being the oxygen and the 2 white balls being the hydrogens. And you can see that it's not written as a straight line. Why is that? Jesse? Mm-hmm. Right. And this is key. So because water is polar in its covalent bonding, so it's like sharing a little bit less with the oxygen is sharing a little bit less of its electrons with the hydrogens, whereas the hydrogens are sharing a little bit more with the oxygen. So that affects the conformation or the steric arrangement of the atoms. And this is really key. The polarity of water is really key for its miraculous functions that it has in supporting life, right? Okay, single bond versus double bond. Which one is stronger? Double bond. Okay, so you have two covalent bonds here in this ball and stick model. It's more rigid. You can think of it as when you think of the difference between an unsaturated and a saturated fat. Those saturated fats are a little more flexible because they lack these rigid bonds. They can pack in really tight and they're solid, at room temperature, whereas like the unsaturated have that little kink from their double bond somewhere in that fatty acid or chain, right? That kind of don't let things pack in quite as well, and so they tend to be liquid at room temperature. So we already went over the polar covalent bonds of water. Let's see, what else? Acids and bases. So the pH measures what? What is an acid on the pH scale? Okay, the concentration of protons are really, it's measuring the concentration of hydronium ion. But there will be free protons in the solution. So so the scale goes from what? 0 to 14. So at 0, do you have a higher or lower concentration? Higher concentration, and that's what we call acids. Okay, and bases have a lower concentration. And really it's just those chemicals in solution will react with the water to form ions. As far as acetic acid will form the acetate ion and hydronium ion. So as a result of when you have acetic acid in solution, it will increase the concentration of hydronium ions, right? Water is what? Just water alone. What pH? 7. So most biological reactions will occur at pH 7 or close to pH 7. So what's the pH of our blood, for example? It's a little higher, 7.4. And that's true of the pH of the inside of the cells, around 7.2, 7.4. So that allows reactions to occur. And enzymes really are dependent on that neutral pH for their optimal function. Okay, so quick review of functional groups. The OH group, oxygen, they're found in alcohols, that's true. What is it called? Hydroxyl. The double bound carbon to oxygen? Carbonyl. Double bound carbon to oxygen with a hydroxyl group? Carboxyl, right? Hydroxyl, carboxyl. The nitrogen? Amino. The sulfur hydrogen group. Sorry? Sulfhydryl, yes. And phosphorus with phosphate group. Yeah. So these are all groups that you'll find, side chains of amino acids. You'll find in biological molecules these groups. So it's important to memorize these groups and know them, be able to recognize them. So in the cell, well, it's mostly water. If we just focus on the chemicals present, or the molecules present, we'll find that most of them are macromolecules. So out of the 30 cell that's made up by chemicals, 23% of it, 4% of it is macromolecules, consisting of DNA, RNA, so nucleic acids, but mostly protein. Proteins are what type of molecules? What functions do they serve in our cells? What type of, sorry? A lot of functions, yeah. So they make up, so can you just give me an example? Structure, yep, they're structural proteins. John?
Some proteins can have enzymatic.
Yep, enzymes. Eduardo? Yep, we got that. Giuliano, did you think of another? Movement, yep. I think you have one more. How do things get from place? Energy molecules are usually energy is stored or found as different macromolecules. So stored as carbohydrates or nucleic acid. Transport. That's what the last thing that I was thinking of. Okay. Transport proteins, channels that are let things in and out of those organelles inside the cell or in or out of the cell itself. So one thing about this, mostly water, what happens to covalent bonds in water? Does anyone know? What happens is, are they stronger or weaker? So basically covalent bonds are much stronger when they're in water, 30 times stronger. And so these molecules are maintained in their proper, what's the really key feature of proteins that enzymes depend on? Really all proteins. They're kept in their proper structure, shape, right? By maintaining these bonds. But there are also a lot of interactions that happen kind of temporarily or transiently in the cell in order to have all these chemical reactions happen that are the metabolism of the cell. And those are also facilitated in the water. So water becomes really important in not only building these macromolecules, but also breaking down, say, the food that's acquired in order to use it to build the macromolecules of the cell. Besides these macromolecules, you also have ions and small molecules, and phospholipids we don't put in that macromolecule category because they're actually aggregates of lipids, not covalently bound polymers that the macromolecules are. So they're a little bit different in their organization. So the macromolecules are all made up of building blocks. Okay, so polysaccharides are composed of sugars. What are these simple sugars also called? Monosaccharides, right? So the subunit that the polysaccharide is assembled from are monosaccharides. Fatty acids make up fats, whether they're lipids or phospholipids as far as membranes. Proteins are a polymer of amino acids, and nucleic acids such as RNA or DNA are polymers of nucleotides. All right. So how do these macromolecules get assembled? They're by condensation, which is the term that your book uses, or I prefer to think of it as dehydration because the name itself gives you a clue as to what it's doing, right? Hydration involves water, right? So these are chemical reactions that use water, and they take out or remove the D water from the subunits to build a polymer. Okay, so this happens when amino acids are joined. So they have a carboxyl and amino end that are joined together to form a what kind of bond? What is the bond that holds the polymer or the polypeptide together? Each amino acid. Peptide bonds, yes. Okay, so in the case of sugars or carbohydrates, you have simple sugars or monosaccharides that are assembled to either into disaccharides, oligosaccharides, or polysaccharides. And these are important. So as I mentioned before, carbohydrates are important for storage of energy in the cell. So can you give me an example of a polysaccharide that might be found in a plant cell? Just the common name for it, Eduardo. Starch. Yep. And we have glycerol in our glucose is what, a polysaccharide or a monosaccharide? Is it a polysaccharide or a monosaccharide? Glucose. How many sugars are in there? It's fructose. Yep. So sucrose is a disaccharide. Do I have this in here? Somewhere. Glucose is a monosaccharide, right? So it's just one ring of carbons, not attached to anything else. But glucose in a polymer can be starch, right? And so in order to build those storage molecules of starch, for example, the monosaccharide through a condensation reaction or dehydration reaction is joined with other monosaccharides in a covalent fashion. So covalently bond to form a disaccharide, oligosaccharide, polysaccharide, which are later broken down by hydrolysis. So the introduction of water to break these covalent bonds in order to bring it to its usable form. Right? So what is the usable form in the cell? Glycerol or glucose? Glucose, what's it used for? Cell respiration, right? ATP, first step. Glucose is a series of steps to pyruvate through the TCA cycle, through the electron transport chain, oxygen being the electron acceptor. You get a whole bunch of ATP produced. So starch and glycogen are those very complex carbohydrates, the polysaccharides. And as we mentioned, for proteins, also carbohydrates in certain organisms can serve as structural components. So where do we find cellulose? Cell wall of plants. Okay. What about chitin? Fungi. Okay. So they both have a cell wall. How about animal cells? Do animal cells have a cell wall? No. So they won't have cellulose or chitin as part of the structural part of their cell. Okay, moving on to lipids. Lipids are common in that they are insoluble in water. Okay, so they have these common traits of being insoluble in water. But this is really actually a key feature of lipids and allows for lipids which also can be a form of stored energy in the cell to stick together in one spot in the cell. It also allows the cell to form barriers between its compartments through the phospholipids, which are spontaneously will form little bubbles within the cell called micelles because of their hydrophilic or water-loving, water-hating parts of the lipids. But basically all lipids are composed of fatty acids, right? They had this hydrocarbon chain that is hydrophobic. It's a long chain of carbons. At the end you have a, what group? It says it right there, just read it. Right? Carboxyl group, right? That's the reactive group that is often attached to another type of molecule at the head, right? So in the case of triacylglycerides, glycerol is what's attached to through covalent bond through that carboxyl group, forming a covalent bond in 3 places with the glycerol. In the case of phospholipids, one of the spots is taken up, so you only have 2 fatty acids attached to the glycerol, and then a phosphate group is taking up the spot of the third, what would have been the third fatty acid tail in the case of triacylglycerides. And this phosphate is bound to a polar group, something like choline, that gives the head or that end of the lipid a water-loving feature. Okay, so you always have that orientation of the head of the phospholipids being towards the water, right? And the tail's kind of hidden inside the membrane, oriented towards each other because of their hydrophobic properties. So these are non-covalent features of the molecules. And this is really important for that function of our cell membrane and all of our internal membranes being selectively permeable, right? Nothing's going to get through here except for very small uncharged molecules. Okay, turning our attention to the majority of what's in our cell, the proteins. These are polymers that are polypeptides, strings of amino acids in a particular sequence. These amino acids all have a side chain that's linked to a carbon. Okay, that's what you call the alpha carbon. And then you have the carboxyl group and the amino group. Okay, and that's how they're oriented in their non-ionized form. In the presence of water, they have their ionized form. And through what, condensation or hydrolysis, amino acids will then be covalently bound together. Condensation, right? The carboxyl group will then react with the, form a covalent bond with the amino group. And that formation of that covalent bond is the peptide bond that forms the backbone of all proteins. Okay? So these are phenylalanine, glutamine, lysine. These are all side chains. And you can see each amino acid has a different structure based on the composition or the atomic, or the atoms that make up its side chain. Okay, so you should go back and we'll look at this again, the structure of proteins. But you should go back and review those 20 amino acids, their names and their structures. So some are polar, some are non-polar. And it has a huge effect on the structure of the protein where these amino acids are found in the sequence. Yes, John?
So do we have to know the name of all the amino acids?
We'll come to that, but you should re-familiarize yourself. And eventually I'm going to ask you to remember the names of the amino acids. So if you learn it now, that helps. Okay, so many unstable conformations of this chain or string of amino acids will occur before the proper stable conformation is formed. Okay, what happens to the function of a protein if it's in an unstable form? Giuliano? Ceases to work altogether. So what is one way that proteins can be denatured? Yeah, Jesse? Heat. Okay. Right, so it's really important that we talked about homeostasis, like maintenance of that body temperature. Our cells are depending on our body to keep the temperature within the limits that the proteins can still maintain their proper stable conformation. That once they're heated, then that change in conformation is irreversible. Yeah. Can be by change in pH as well. Yep. So pH, temperature are major factors that will affect the function of enzymes. So conformation doesn't only depend on these covalent bonds or the peptide bonds between the amino acids in the protein. The conformation depends on non-covalent interactions, including electrostatic or charged interactions, and hydrogen bonding, and van der Waals interactions when they're in in the case of the quaternary or multi-subunit proteins. Okay, so turning to nucleic acids, which are composed of some units called nucleotides. The nucleic acids come in two forms, the DNA and the RNA, having a different base. So ribose and deoxyribose being the difference in the sugars. And the nitrogenous base, one difference between thymine and uracil. Uracil found in RNA, not in DNA. So it is the phosphate group that is reactive that forms a covalent bond with the next sugar. Okay, so the carbon in that covalent bond between phosphates is called the phosphate backbone of DNA. And remember that DNA doesn't come as single-stranded molecule, right? It's double-stranded. So what's holding those strands together? Hydrogen. So even though those are weak interactions, they hold the double helix in a stable conformation because of the base pairing. So which bases pair with which? Guanine, cytosine, adenine, and thymine. So basically purines with pyrimidines. Okay. Okay. And which one has how many hydrogen bonds are formed between guanine and cytosine? 2 or 3? We have 2 votes. 2 or 3? Okay, vote for, those of you who vote for 2, raise your hand. 2 between guanine and cytosine. And those who believe it's 3 hydrogen bonds between guanine and cytosine. Gee, most of you are abstaining. Come on. Okay, so it is 3. So How can you tell? Just look at the hydrogens that are available between the guanine and cytosine. Okay, 1, 2, 3. Whereas here, there's only 2, right? Hydrogens have to be involved in hydrogen bonding. If you don't have a hydrogen bond there, then you won't have a hydrogen bond. If you don't have the hydrogen ion there, you won't have the hydrogen bond. Okay. So one other nucleotide that I want to bring your attention to and have you refresh your memory about is ATP. Okay, is adenosine triphosphate. What is ATP in the cell? What function does it? Alex, yes, it's the energy currency of all cells. Yeah. So what makes it the energy currency of all cells? What part of the molecule, Juliana?
Phosphate, the third phosphate.
Yeah. So actually the second and third are unstable bonds that can be easily broken to release energy that's necessary for catalyzing all of these chemical reactions that are happening in our cells. So ATP is really key to the function of our cells. Without ATP, we would not be able to capture that energy that is necessary for life. And without enzymes to catalyze the reactions, there would be no life. Okay, so these two final phosphate groups are linked by phosphoanhydride bonds. And ATP is constantly recycled. So once it gives off the energy that's used for cellular work, it's converted to ADP. And then whether it's a photosynthetic cell or a chemotrophic cell, the cells are able to phosphorylate ADP and return it to its high energy status of ATP by adding inorganic phosphate in one of 3 ways. Substrate phosphorylation, oxidative phosphorylation, or photophosphorylation. Okay? Okay, finally, looking at these weaker interactions that are really actually key for the function of molecules, these macromolecular interactions. Yeah?
You said substrate phosphorylation.
Oh, okay, let me write those down. So the ways that phosphate group is added to ATP is through substrate phosphorylation, oxidative, or photo phosphorylation. Okay, so these two happen during respiration. This one happens during, so substrate phosphorylation occurs during fermentation and respiration. Substrate and oxidative phosphorylation occur during respiration, and photophosphorylation occurs during photosynthesis. So a lot of what we're going to be talking about of the inner workings of the cell involves the interactions of molecules or macromolecules. And these interactions, whether they're a ligand with its receptor, an enzyme with its substrate, they're dependent on these non-covalent interactions of electrostatic interactions, hydrogen bonding, or van der Waals interactions to facilitate the interaction long enough so that the function can be achieved. And one example that's given in your book is that beyond the macromolecular synthesis by condensation of amino acids to peptides or nucleotides to RNA, the ribosome then uses these peptides or takes these peptides and the RNA and combines them into a functional organelle by macromolecular assembly. Okay, so the covalent bonds that hold these macromolecules together are not enough to make a functional ribosome. In addition to that, you need to have the non-covalent bonds of hydrogen bonding and van der Waals interactions that occur to bring the subunits, each of which composed of both peptides and RNA together into a functional ribosome, so that you can get, and here in this electron micrograph, every little black dot is a ribosome sitting on a strand of messenger RNA. Okay, so to get those proteins produced from that message. So some of what happens in the cell is simply by random diffusion. So what happens to molecules when they're in a closed environment? Where do they tend to move? How do they tend to move? If you have two substrates, A and B, how do they tend to move? Yeah, Alex? Just randomly, but they also might have a directional movement based on what? Juliana? Concentration. So if you have an area where there's a lot more A than B, the A will tend to move to an area where there's less A until you're reaching that state of equilibrium.
They tend to move in the direction based on what?
Concentration. So if you have more of A in one part of the cell and less in another, it will tend over time to even out. And as shown in this diagram, the more of those non-covalent interactions that occur between two molecules, so the better fit, we could say, that they have, the more likely they're going to stay connected. Okay, so if the enzyme, think of enzyme substrate, which we'll talk about when we go over chapter 3. Enzyme substrate, the key is that shape of the enzyme has to match the shape of the substrate, has to have the right side chains to allow for hydrogen bonding to occur, van der Waals interactions to occur, so that the substrate can bind the enzyme long enough to be converted to the product, right? So even with multiple weak bonds, they can stick together long enough to achieve their function, these molecules within the cell. All right, so here are the objectives for chapter 2. Just going to check what time it is. Yep, so our time is up today, but I've gotten through chapter 2. Take note of the objectives when you're reading chapter 2 and taking notes. I do expect you for the exam to be able to draw an atom. So remember the nucleus with the electron cloud, as well as the water molecule, including its polarity. So remember which part is electronegative, which is electropositive. So go ahead and review this in preparation for that. And again, I encourage you to try the Chapter 2 quiz. And I've selected some problems. Remember the problems in your book, you can also check your work because the answers are in the back.